Hello, everyone. Thank you so much for joining us for the sixth edition of the GSU MalariaGEN Journal Club monthly series for 2025. Today's webinar is a parasite-focused one, referencing Dr. Nina White's et al. It was a group effort. A lot of people were involved. Recent paper, which is titled Pf8, an open data set of Plasmodium falciparum genome variation in 33,325 worldwide samples. My name is Pablo Chauke, for those of you that do not know me, and I'm a training coordinator at the GSU, working with a team of awesome people, some of whom are in the room to organize these monthly events. Just a few ground rules. Our journal club is being recorded and will be available on the MalariaGEN YouTube channel so that you can rewatch, relearn, share with your friends, colleagues, and networks who couldn't attend today. Please stay on mute so as to not disturb our speaker, Dr. Nina. We'll have a Q&A session at the end where you can either raise your hand or type in the Q&A box or the chat box as well, and I'll read them out for Nina and she'll respond and engage with you further. Without further ado, I would like to reintroduce our speaker for today, Dr. Nina White, who gladly agreed to give our sixth journal club for 2025 when I invited her. For those of you that attend our GSU Journal Club series, you might have met her previously in 2024 when she brilliantly co-presented on PF Upload Atlas app with Chun. You can find that on our YouTube channel as well. It's one of our most watched videos as well. So from experience, you know this is going to be a great talk, no doubt about that. The title of the talk is going to be similar to the paper, which is about Pf8 dataset. But just to remind you about who Nina is, Dr. Nina works with the malaria parasite surveillance team in the Genomic Surveillance Unit, GSU for short. And in that role, she helps process, analyze, interpret genomic information from malaria parasites of the genus Plasmodium. She also supports our partners to do similar work across the world. She also researches and develops ways to improve the team's service as well. This work that she does helps to track the evolution of the parasite and in turn supports public health decision-making to better mitigate malaria. Dr. Nina White, thank you so much for joining us. You may begin your presentation.
And thank you, Pablo. All right, I'll get started. Okay, so yeah, I'm Nina and I'm a genomic data scientist in the parasite surveillance team here at the GSU, part of the Wellcome Sanger Institute. And I'll be introducing Pf8, which is the latest release of Plasmodium falciparum whole genome data from MalariaGEN. And it's important to state that while I've got the honour of presenting today, this really is a community project, and Pf8 is possible thanks to the work of all of the MalariaGEN network. So really, I'm presenting on behalf of them. So I hope the presentation should take around half an hour today, and it's split into two halves. So the first half, I'll cover what's in Pf8, how we made it, and some new features as well. And then in the second half, we'll move on to a kind of a walkthrough of the apps and tools which are released alongside Pf8 to hopefully showcase how to get the most out of Pf8 in terms of how to access, interpret and analyze the data as well. So the Malaria Genomic Epidemiology Network, MalariaGEN, has been collecting and curating whole genome data from the two most important malaria species, Plasmodium falciparum and Plasmodium vivax, and making these data releases which are publicly available. Pf8 is we think, the largest non-human eukaryotic whole genome database. And the vast size and open nature of these releases are really significant for enabling malaria genomic surveillance and research to be done worldwide. And we've had many publications citing the resources, which is really great. And today I'll be walking through Pf8, which is the next iteration on from Pf8. So for Pf8, we've expanded on Pf7. We added around 12,500 new samples, bringing us to 33,325 whole genomes. These come from 34 countries and 122 locations within those countries. So that means the first level administrative division, and that's an expansion of 23 locations. We've also expanded our network of studies for Pf8 compared to Pf7. So we're now at 99 studies, so just shy of triple figures, and that's added an extra 17 studies for Pf8. We have samples in Pf8 that span as far back as 1966 and they go to as recently as 2022. So there's over half a century of genomic information contained within Pf8, which is pretty cool. And with the extra samples that we've added, we have found extra genetic variation relative to Pf7. So contained in Pf8 is around 12.5 million genomic variants, and that's either single nucleotide polymorphisms or SNPs or insertion deletion indels. And with a genome size of 23 megabases for Plasmodium falciparum, that means there's variation in over half of the genome in Pf8. So as Pablo said, the publication for Pf8 is now out, and you can find that on Welcome Open Research. You can scan the QR code to get to it there and access the paper, the methods, the figures, and also the supplementary information to go alongside it. So yeah, we encourage you to go and check that out if you haven't already. So now I'll talk a bit about what went into making Pf8. So we ran a variant calling pipeline for all of the 33,000 samples to discover SNPs and indels across the genome. We apply some quality filtering within that pipeline to the genetic variants, but then we also run quality control on the samples after that, taking into account their metadata. So this means we remove or we QC fail samples which show evidence of a mixture with different malaria species to falciparum, any samples which we think have contamination or are a lab strain, or samples which have mistakes in their metadata. For example, the continent was mislabeled. And after quality control, we have 24,409 samples which pass QC for Pf8. And it's this subset of QC pass samples which we have taken forward to perform a selection of analyses which are described in the paper. And these fall into sort of three broad categories, the first of which is population structure. So the figure below shows that in Pf8, and the same was found in Pf7, in that the genetic structure resolves sort of most sensibly into 10 global subpopulations. So going from South America through West, Central, East and Northeast Africa, to the east and far east of South Asia, both the western and eastern halves of Southeast Asia, and also Oceania and New Guinea as well. And if you check out the paper, you can see some neighbor joining tree, principal coordinate analysis, and using FWS to describe the clonality of samples as well. We have also run all of the samples through a copy number variant calling pipeline to discover CNVs. So that's amplifications in key genes linked to drug resistance or deletions in the HRP2 and 3 genes, which are linked with rapid diagnostic test failure. And we describe that per population. And we also take the CNV information and mutation information at key genes, and we produce drug resistance phenotype predictions for all samples as well for some key anti-malarial drugs, which is obviously important from a public health perspective. So that's a recap of Pf8 and what was sort of similar to what was included in Pf7. But there are some new things for Pf8 as well. So first off, we have created a SNP-only call set alongside the full call set, which contains SNPs and indels. And we decided to do this because we know that often for analyses, it's common that you might want to just select SNPs or biallelic SNPs because indels can be quite tricky to work with. But we noticed that with the extra samples, we were discovering more rare indels. So positions in the genome which previously were just a SNP were now a SNP and indel. So to pull out the SNPs, we thought would be a good idea. So we removed all the indels, sort of unmasking those SNPs. And this call set is available and has 10.8 million SNPs for analysis. For Pf8, we have open sourced both the variant calling and the copy number variant calling pipelines. They both each have a GitHub repository, and these are linked from the manuscript as well, so you can go and check those out. And we decided to produce copy number variant calls for two new genes in Pf8. These are GCH1 and CRT. So GCH1 has been linked to antifolate resistance, and CRT, obviously mutations in this gene are linked to chloroquine resistance. Amplifications, not so much yet, but we thought that this would be useful information to provide as it is an important gene. And the figure shown on this slide just shows some patterns of interest we saw for GCH1 amplifications. So we see quite a marked difference in their frequency over the past couple of decades in the western half of Southeast Asia. So that's this light blue line, and in the eastern part where it's much lower, but in both has shown a small decrease with time. And this sort of contrasts what we saw in West Africa, so that's the red line, which in the most recent years we have data, we saw a modest increase. So that's kind of a recap of some of the analyses we did, and we decided to showcase a few examples of analysis that could be done, but we stopped short of trying to do everything for the paper, and really that's because we think that the value of these data resources comes from the power of what the community does with them, so many brains put together to make innovative use of the data. And to that end, we release a set of apps and tools to allow the raw data to be worked with, and that is what I'm going to spend the rest of the presentation talking about. So walking through all of these different apps and tools to hopefully give you an idea of how to get the most value out of Pf8. So this is a map of where we'll be going. So we'll work through from the left to the right across the two apps and the two types of tools we have. And the sort of pattern to this is that we go from the highest level overview of the data, and with each step, the tools bring in more genomics, more coding, and ultimately become more customizable to your own needs as well. So I'll start by running through the Data Explorer web app. This video will just play in the background. The quality of the video should improve, but essentially this is a web app which provides a very high-level overview snapshot of Pf8 with an easy-to-navigate dashboard. So it showcases quite intuitively where the samples came from, how many passed QC, etc. And you can start to drill down to different levels. So here we've got all of the countries in Pf8, the number of locations which were sampled in each, the number of samples, and the time span for each country as well. And then if you are interested in a specific country, so for example here, the video is showing we've clicked on Cambodia, we get a country level overview, but then also an overview for each of the locations within that country. And a new graphic that's popped up here is this one to the right, and this is drug resistance predictions for all the samples in these locations, so for 6 different drugs, and whether the samples were resistant, sensitive, or we couldn't make a call. You can also view this information on a study-by-study level, again, showing the same information but just sliced slightly differently. And finally, you can get to a per sample view and apply different filters as well to get to the information you're interested in. And yeah, we have drug resistance predictions for each individual sample as well, which may be useful. The web app also provides links to important publications, to the people who've contributed data, and just some extra documentation as well. So if you scan this QR code, you can check it out for yourself. And just to sum up, the Data Explorer app is a simple and high-level overview of Pf8, displayed in an easy-to-navigate dashboard. And you can view drug resistance predictions in various different summaries as well. So that's good for a really high level sort of snapshot of the data, but what it doesn't show is any genetic information. So if you are interested in the mutations that are underpinning these drug resistance predictions, or if you're interested in a gene that isn't linked to drug resistance and you have another gene in mind that you'd like to look at, we'd recommend looking at PF haploatlas. So as Pablo said, we did run a journal club on PF haploatlas, but so you can check that out if you would like a more detailed run through of it. But essentially PF haploatlas is an app in which you can explore haplotypes through time and space for any of the genes in the core falciparum genome. So that's almost 5,000 genes and haplotype information based on the Pf8 whole genome data. So there's a brief description on the landing page here, and essentially to get started, all you do is go to this search bar and either look for a gene, type in a gene name that you're interested in, and pick one from the list. So in this example, we're looking at Kelch13, which is linked to artemisinin resistance. And what you'll get then is a summary of all the haplotypes which had more than 25 samples, which is just the default filtering, but you can change that. And each haplotype is shown vertically, and haploatlas will tell you how many samples had each haplotype, where each of these haplotypes were found across the 10 global populations, and then what mutations are found within each haplotype. So here we're highlighting the C580Y mutation in Kelch, which is pretty infamous. And by clicking on that, we're then taken down to an even more detailed view. So here we have first level administrative divisions on the y-axis, years going across the x-axis, and the intensity of the colour of each of these dots represents how frequent that haplotype was. So we can see a big cluster of high frequency C580Y in Southeast Asia. And finally, this information for the C580Y haplotype you can view, but aggregated at the country level with a customizable time span as well. And all of these images are downloadable in various formats. So hopefully you can get to publication ready figures easily. We also provide the ability to download the raw data aggregated at the population level or at the sample level if you'd like to take this data and you have in mind a different analysis you'd like to do. You can also explore the gene further on Plasmodium by clicking that button. And I think here we're just showcasing, a different gene, CRT, linked to chloroquine resistance, and showing example if we lower that minimum number of samples filter, you can see the number of haplotypes increases as well. So you can adjust this to your own interests, and this shows what a haplotype with multiple mutations will look like on that bottom plot as well. That's just running through the rest of the plots. There's a demo video embedded, some further descriptions of each of the plots, links out so that you can provide feedback, and a nice section showcasing some quotes we've had from users of PF haploatlas, which were really nice for us to get. So to sum up, PF haploatlas is our web app that allows you to explore haplotypes from any of the 5,000-ish PF genes through time and space, and you can download raw figures and data as well there. And scanning the QR code will take you to the app. Just bear in mind that it is probably best viewed on a laptop or computer rather than a mobile for now. So going forwards from haploatlas, we'll move into what we'll call tools. So now I'm going to show what are public notebooks, which are a Jupyter notebook which hosts interactive code And these notebooks provide accessible and guided analysis examples for MalariaGEN data. And we call them public notebooks because they run completely off of publicly available data resources, cloud stored data using our Python package, and the computational resources come from Google Colab as well. So there are a set of examples in which all you need are an internet connection and a computer to start analysing Pf8. They are all hosted on a GitHub page, which will be linked in the next slide, and summarized here into sort of 4 broad categories. So useful genomic how-tos, just some useful procedures for working with genomic data, ways in which you can summarize Pf8 data with tables and figures, and a set of analyses which deal with evolution against malaria control and population and structure as well. So we're going to look at one now, which deals with drug resistance trends in Africa. So with just one click, you're taken to a static rendering of the code, but these really become interactive when you choose to launch it in Google Colab by clicking on that rocket icon there. And this is what the notebook itself looks like. So there are chunks of code here, which you can execute by clicking on the arrow, and then the code will run and sort of come to life. All of these notebooks are broken down into step-by-step instructions, and we hope well annotated enough so that they guide you through and provide you with ideas for how to perhaps customize the code and take it forward into your own analysis as well. So this is just showing the beginning of this notebook and the steps taken to process the data. And ultimately, we're going to come to a map of chloroquine resistance in West Africa. So in Haploatlas, we were just looking at CRT, which is linked to chloroquine resistance. And this is an example of how you can expand on that analysis. So here we have a map of West Africa and chloroquine resistance frequencies among different countries between the years 2018 and 2022. So from this, we can see that in Ghana, levels are actually very low relative to the region, and in places like Gambia, especially here in Western, the majority of samples were chloroquine resistant. So that map was something we had for PF7, but we've also expanded on this notebook and provided an analysis of chloroquine resistance through time for 5 different countries. Oh, excuse me. And here's an example of if you are interested in a different drug, you can go ahead, type in sulfadoxine, rerun the cell, and then the figure will change. So now we're looking at sulfadoxine resistance over time in these 5 countries. And as you can see, it has been increasing for all of them. So now in this video, we'll just jump through a couple of other public notebook examples. This one I like because it's in the useful genomic how-to section, and it uses IGV notebook, so that's interactive genome viewer. And I like it because it allows you to get a look at the raw sequencing reads, which I find can be a little bit abstract to imagine sometimes, but this shows you essentially the reads from the sequencer aligned to the reference genome, which is really useful if you want to visualize things like gene deletions. So here we're looking at a sample which has a deletion in HRP2, and I think you'll see that there are reads and then they disappear, which indicates a deletion. An IGV notebook is nice because it gives you, on hovering over the reads, you get it's interactive and it will give you information on that read's characteristic, like its mapping and etc. So if you want to scrutinize a mutation or a CNV, this notebook could be a nice thing to use. And I think just quickly now, we'll look at one example of population structure analysis. So here we create an interactive neighbor joining tree to look at the different populations in PF8. So yes, we encourage you to go and check all of these out and let us know what you think. And just to sum up, these are interactive guided analysis examples using open data and cloud compute power. And the code should be customizable for your needs. So finally, if you have your own pipelines or your own code ready to go, and really all you want to get to is the raw data, the next slide will walk through how the different ways you can do that. So here we're showing the resource page, which will be linked on the next slide. So this provides links to all of the apps you've just seen and some other useful reading. But also here we have raw and derived data sets. So we've got GVCF files, CRAM files, and derived data sets such as genetic distances, and all the SNP-only call sets I was talking about earlier. There's a README file which goes into detail as to what's contained in each, and also includes some instructions as to how to work with VCFs to extract certain variants. And all of these data are completely free to download, stored in an S3 location. I think you can see here, you might look at the VCF files and you can get to all of those if you wish. But if you don't want to download really large files, there's over 100 terabytes of data making up Pf8. We would recommend you go back to this GitHub page and check out the PF data access guide, because this tells you how to use our MalariaGEN data Python package to access the data from the cloud with no need to download it at all. And you can get to the metadata, the variant calls, genotypes, and annotations as well here. It's just being run through in this video. Okay, and yeah, if you scan that QR code, you should get to the resource page. But yeah, we provide access to raw and derived data sets completely free for you to download or to access with the MalariaGEN data Python package. So that concludes our walkthrough of Pf8. I really hope that you found that insightful and useful, and we encourage you, to go ahead, use it, but also please let us know what you think and how we can improve going forwards as well. And just to sum up, Pf8 is an invaluable foundational resource for understanding evolution. And it provides over 100 terabytes of data and dedicated apps and tools for free for the community as well to combat malaria. Going forwards, we are already working on Pf9, and a key development here is that we're hoping to start bringing in all publicly available whole genome data. So previously we just have data which has been sequenced with us at the Wellcome Sanger Institute, and we hope that this might bring the size to over 50,000 samples, which would be a great milestone to hit. And we will also work on PV5, which is the next resource for Vivax. And that just leaves me to say thank you to the entire MalariaGEN network for contributing data and collaborating on Pf8, to all the teams within GSU who've also collaborated to produce Pf8, and to our funders at the Gates Foundation as well. So thanks very much, and looking forward to any questions you might have.
Okay. Thank you so much, Nina. That was fantastic. I did say in the beginning that it's going to be amazing as usual, so well done. Congratulations to you and the team. I think this was a massive mammoth task. It's quite impressive, so well done to you and everyone, all the partners involved as well. We've now reached time for Q&A as well as discussion. Please type your questions in the Q&A box or raise your hand, and Nina will be able to engage with you. I see there's already a hand from Temitayo Ogidumi. Please go ahead and ask your question. I'm going to allow you to talk now. Please unmute and ask your question.
Good afternoon. Thank you, Dr. Nina, for your detailed discussion about PF8, and thank you for putting it out. I was actually working on PF7, and I was excited about what I was doing. And when PF8 came out, I was like, "Yo, what is going on? I'm not even feeling what I was doing with PF7. Why is PF8 out already? And it was very exciting for me and my supervisor to see that. But I've been trying to access the data, and the platform I'm using, the server I'm using is about 232 GB RAM. And what I'm trying to do, I need to get the the genomic features out because I'm trying to build a machine learning model to do some things. So I'm now trying to see if I can extract a specific amount of samples because it's a continuum of PS7. So I would just want to remove just the new additions, which is about 8,000 sample IDs. So I was trying to do that yesterday, but it was failing. I can't recall what it was telling me. So I
Mhm.
don't know if you have an insight into what I can do to help that work. So I was working from the Google Cloud end. So I had a script that is using the Google Cloud. So it's not loading it directly into my server space. It's just calling it directly, but it is failing. So I don't know if you
Mhm.
have an insight around that. Thank you.
Thank you. Really glad to hear you're excited about PF8. For that specific problem, yeah, I think we've found that working on the cloud infrastructure, sometimes there's just too much data for the available RAM. But I think probably the best thing to do here, if you don't mind, if you could write that up in an email and send that question over to me with the error message that you're getting, I think we should be able to hopefully help you a bit better that way as well, if we have it in writing.
Oh, great. Please, what's your email then?
I will post it on the chat now.
Oh,
If I
thank you. Thank you.
get to it.
Okay, thank you so much, Temitayo, for your question. Nina is going to share her email to everyone in the chat box, and you can email her. It's nw20@sana.ac.uk. Thank you so much, Temitayo. We have another hand from Jocelyn Guerrero. Jocelyn, please go ahead and ask your question.
Hi everyone, thank you very much for this talk and for introducing the new feature of Pf8. I wonder if, because I have been reading the pipeline that you are using for calling SNPs, I wonder if there is a way to get the recalibrated BAM files before you are just doing the selection of the 10 million SNPs that you are now providing.
So just getting to the raw BAM files, is that the?
Oh, the recalibrated BAM files.
Could you explain just what you mean by the recalibrated BAM files?
So in the first mapping, you get the BAM files, and later I think with upload type color, one of the tools of GATK, you get a second that when you verify a verification of the SNPs.
Okay, yeah. So I think what we provide is after the second stage, the CRAM files that are available would be there, and I probably need to go away and just check on the recalibrated BAM files as to whether we have those and whether we could share them. But at the moment, I think all we have released is the BAMs after that second stage, I'm afraid.
Thank you very much, and I'm very excited and looking forward to see the PF9.
Great, thank you.
Thank you so much, Jocelyn. I like how everyone is also now already looking forward to PF9 after this huge task of doing PF8. I've shared the paper in the chat, but I'll also shared Nina's email as well. I see there are questions in the Q&A. There's a question. Okay, so Chun says, also feel free to send emails to support@malariagen.net. So that's from Trin. Thank you so much for that additional email that participants of this journal club can contact. And Richard is also sharing the CREM files, where the CREM files are available that are recalibrated. Jocelyn, I think that's for you. If you check in the Q&A, but I'll also copy this and paste it in the chat. That's in response to you, Jocelyn, from Dr. Richard Pearson. We still have time. We have about 25 minutes to go, so we still have time for more questions, for more discussion as well. So please do raise your hand or type your questions or points in the Q&A box. And Nina will gladly respond to you. And it's so lovely seeing so many familiar faces. I know I'm not seeing your faces, but familiar names in the participants list. So thank you so much to all of you for joining. Okay, questions are coming in, or other responses as well. Dr. Richard Pearson says, raw data before any calibration can be found at the European Nucleotide Archive, or ENA for short. There's a question from Antoine Dara. How do you see the future of this resource in 10 years? Nina?
Yeah, that's a great question. I think ultimately it is going to become even more so a community effort to produce and to contribute to as well. I think there's an important step forward for Pf8 in terms of starting to bring in data that has been sequenced outside of the Wellcome Sanger Institute. And I think that in 10 years' time, we hope that that these resources are something that are built really, even more so in a community way, you know. So at the moment, we have a big hand in running the pipelines and curating the data as well. But I think we really, I think the vision hopefully is that it will become something that is more decentralized. Yeah.
Thank you so much, Nina. There's a hand from Abibi. I hope I'm saying your name right, Abib. I'm going to allow you to talk now, so please raise a question or comment.
Can you hear me?
Yeah, I can hear you.
Yes, we can.
Great. Nice talk. So one of my questions someone already raised, because like maintaining this resource is very important. So I'm thinking 10 or even after 10, 15 years, how to maintain this one because of this, the funding dynamics currently we have. And then I have two follow-up questions. So the first one is, I didn't look at closely the paper, that's my to-do list over weekend, but which version of JTK pipeline you guys use to do this analysis? One of the question. And the second one is really very important, that any of the population structure or whatever the population genomic analysis you guys doing, because you show there are a lot of samples temporal components. So when you show like the parasite Ethiopia is different from Kenya, or Southeast Asia different from Papua New Guinea versus Solomon Island, how are you controlling the temporal component on those analyses? Because if you show those things, I mean, the structure we're seeing, is that really the true structure, or is that due to the temporal component? Thank you.
Thank you. So you're testing my memory there with the GATK version. I'm hoping maybe Richard or Chien could post that in the chat if they have access to the paper now. And then that's a really good point about the population structure, because we're showing it sort of static, zoomed out, disregarding time from that neighbor joining tree that you would have seen. So it would be interesting to look at that broken down across different time points as well, to see how that is changing, if at all. I think, broadly at these larger subpopulation levels, there are differences that are the parasites are quite deeply diverged on, between Southeast Asia and Africa, for example. Maybe wouldn't change so much with time, but I think you raise a really interesting point there. Oh, and Chien has just posted in the chat, GATK 4.2.5.
Oh, great. Thank you.
Okay. Thank you so much. Also loving the teamwork in the background. Dr. Richard Pearson and Chien coming in as well. Thank you so much. Thanks, Abib, for your question. We still have time, as I said, about 20 minutes to go. So please do raise your hand. I'll allow you to unmute and speak, or you can type in the chat or Q&A box as well. Nina, as I think I looked at the paper and there was about like more than 40 authors. So there's a lot of people that are involved in the background. Any reflections on the amount of work and effort that it took for Pf8 to come out and the complexities, challenges?
Yeah, I mean, yeah, it is a massive, massive effort in terms of that becomes really clear when we are writing the author list. I mean, that's quite a task in itself, filling that in when we're submitting the paper, and that's shout out to Julia and Ishla for their work on that. Yeah, it's a massive effort and has been a massive sustained effort for decades really with MalariaGEN and curating these. So yeah, an absolutely huge amount of work. Yeah.
Thank you so much for that, Nina, and well done to everyone involved as well. Quite impressive. We have about 2 questions currently in the Q&A. There's a question from Peter Morris. Thanks, Nina, for the excellent talk and everyone in the team for this impressive accomplishment and your commitment to open science. I have a question about the CNV pipeline. Can the HMM training data of Pf8 be reused for calling new samples?
So I am going to defer to Chun for that, who really was the mastermind behind the CNV pipeline, if that's all right, Chun. Perhaps he can type an answer to you.
Chun, I can also allow you to unmute.
Oh yeah, that would be.
Let me go ahead, Chun. I've unmuted.
Hello, can you hear me?
Yeah, we can hear you.
Cool. Yeah, hi Peter. Yes, I know you. We've been talking about CNV calls in the past, but we have engineered it in a way where, yeah, you can sort of make CNV calls on just 1 or 2 samples. So yeah, we should be able to sort of get something arranged. If we email, we can sort of discuss what's the best way to get that done.
Okay, thank you so much, Chun. And the email has been shared, both Nina's and the support at MalariaGEN.net, that you can email as well, Peter. Thank you so much for your question. There's a question from Anonymous. Please, I have difficulties genotyping short tandem repeats for PF using some tools like HIP-STR, Gang-STR, Ensemble, Expansion Hunter, since these tools do not have the bed files which contain short tandem repeats from the Plasmodium parasite. Please, do you have the bed files for Plasmodium, which I can use with these different genotyping tools to call the short tandem repeats from Pf8 dataset? That's from Anonymous. Nina?
So that's something I would encourage you maybe to put that in an email to either one of the email addresses we've shared, either support@malariagen.net or to one of our team, just so we can definitely get the specifics of what you're asking for in writing, if that's all right with you.
Okay, thank you so much, Nina. So yes, please use the email. The team will definitely respond to you as well. There's a question from Catherine Bakari Mva. Hello, nice talk. So for CNV, which tool does the team use, or is it just based on alignment with IGV? That's from Catherine. Nina?
So I say broadly we use 2 methods based on coverage and based on identifying breakpoints. But again, I think Chun is probably better placed to give you a more detailed answer there, if you wouldn't mind, Chun.
Yeah. So as Nina said, we're largely using 2 methods. One of them uses a string of GATK tools such as GCMV, germline CMV caller, and yeah, all the methods are sort of openly accessible. The code base is fully openly accessible, so I might refer you to the paper. But also we have a sort of custom method which looks at the frequency of face away reads, which are reads in a very specific orientation, which is different to normal, which gives us clues about tandem duplications. again, all methods very well documented in the paper and the repository. But if you have any further questions, again, feel free to email us and we'll be happy to help.
Okay, thank you so much, Chun. A second question from Peter Morris. You've spent a lot of effort into making your data available. It says not available, but I assume it means available, but also easy to interact with. Any important learnings or tips for other research teams who also want to go down this path? Nina?
Oh, that's a good question. I mean, I think the answer really is asking and getting feedback on how people want to interact with your data. I think, to be honest, I think we could still do better at doing that. But since releasing especially Haploatlas, for example, and the public notebooks too, we're always trying to gather feedback on what people liked, what they didn't like, so that we can improve always. But yeah, I don't know whether Chien or Richard, you have any thoughts on this as well, because that's a good question.
Yeah, I could go first. Yeah, I think as Nina said, we try to interact with our users as much as possible. At conferences, we'll we'll set up questionnaires to understand like what is it that people exactly want. Quite often we've been we'd actually learn quite a lot about, we thought maybe people wanted tools mainly, but in fact people want a variety of sort of learning and coding support and workshops, as well as tools that help people to navigate the data more easily. So yeah, I think it's just really understanding sort of the customers.
Okay, thanks, Chun and Nina for your response to that good question. We still have about 12 minutes to go. So if there are any more questions, I'd encourage you to, again, type in the Q&A or chat box or raise your hand and I'll allow you to unmute. And just to remind you, this is being recorded and will be available on our MalariaGEN YouTube channel. You can rewatch anytime as well. There's some email addresses that we've shared and some links to the paper as well as the apps and tools as well that you can look at after this Journal Club. Dr. Pearson says, given the size of the data, we have found making available on public cloud resources is quite helpful as no large data downloads are required. Peter, I think that's in response to your question about any advice to other teams as well. That's from Dr. Richard Pearson, who's the lead of the parasite surveillance team within the GSU. So thank you so much for that, Dr. Pearson. Okay, any other questions or comments before I let everyone go? Going once? Going twice? Okay, well, going 3 times. Thank you so much, everyone, for joining the July Journal Club. We are going to have a next Journal Club on the... Okay, there's a question before I say goodbye. Antoine, we'll let you speak. Let me unmute you and you can ask your question. Or was that a mistake? Because I see the hand is down now. Antoine? Okay, I think that might have been a mistake. Okay, so we are going to have the next Journal Club on the 19th of August. And our speaker is actually in the room right now. Balotin is going to be presenting on his recent paper as well. So looking forward to that. So kindly look out on our social media pages such as Blue Sky, LinkedIn, our website, our newsletter for that announcement as well. Nina, as usual, you were fantastic. Brilliant presentation as usual. Thank you so much for your time and engaging with us on the new resource. And well done to the MalariaGEN, GSU teams that are based at Senga, but also globally as well, because I think, as you say, this was a collaborative team effort. And I'm looking forward to seeing what you're going to do with Pf8, as well as you've done amazing things with the previous releases as well. So well done to you and to your team and everyone who was involved in making this a success.
Great. Thank you,
Any
Babalo.
last comments from you?
No, just thank you. And thank you to everyone for your questions.
Okay. Thank you so much, everyone. Have a good day and see you next month.
Bye.